we have seen how to
You open how to download it.
okay. So I was saying you just click on download. Eh?
But the advice,
Okay, but have you known how to do you know do you did you follow up how to download it? Hello? Oh. Hello, can you hear me? So I was saying
yeah, I
that
can hear you. I can hear you.
I was saying that you always go for 3D structures instead of 2D structures, and if there is no 3D, then download 2D. But after downloading 2D, you must use your CameDraw to convert the 2D into 3D. So how to just download it? You click on save SDF. SDF means structural data file. So you just click on the SDF, the save under it, and the structure will be downloaded. Okay. So then you cancel it. You go back and type in another. You type in another structure of your choice. So that means part of the things we'll do now is we are going to exploit our capability in downloading chemical structures. So I will give an assignment after today. I will assemble some structures, and I will send them to you so that you can see how you can be able to download the Pubchem, their structures in SDF. Although downloading structures when dealing with natural products is always stressful, yes, it's always stressful, but we are going to practice it because I could remember when I learned chemoinformatics, the first assignment that was given to me was to download about 27,000 compounds in less than 1 week. So I was subjected to download 27,000 compounds within a week. I had no choice. It's part of the task because it will help to build us in structural to retrieve data. So it's part, but I'm not giving you 27. No, I'm not going to give you 27,000 compounds. I'll just give you small, like 500, like 500 to download. I'll provide their names, and you see how you can practice it. Okay, so outside downloading
You say
structures,
like how many, sir?
like 500.
Okay.
Uh, uh, 500 is not even up to 1% of 27,000. So
More than a punishment.
uh, and it's the task that comes with it. If we don't do it, it now mean that we are joking with structure retriever. Okay, so we have seen how to download exact match, downloading your structure. Let me use another compound, camphorol. Now let's say we have another compound, camphorol, for instance.
Yeah.
Let me now show you how to do substructure. Now let me say my target now is not downloading structures in my plant material, but my initiative is to work on compounds that are related with camphorol. Compounds that has that camphorol structure. Maybe the difference might just be some substituents attached to it. Maybe let's say that camphorol was effective against ulcer. And if you can have some compounds that looks like camphorol, that means they might exhibit better or less than the camphorol. So in that case, the kind of approach you are going to use is similar structure search. You are going to use similar structure search. And let me come down here. Here, permit me now to explain this. Under compounds, we are seeing 625. That means there are 625 compounds that has camphorol as part of the name. Yes. Then there are 3,116 substances. These ones now, they are not compounds. Let's say like antibodies, like hormones that are related to camphorol. Here, there are 19 genes. When we get to, maybe when you'll be coming for the training on bioinformatics, remember I told you that you can get any information related to a compound in chemoinformatics. So yeah, we can get the genes. We can use Pubchem to get the genes, those genes that are associated with camphorol. So once we click on genes, what will be seen is the genes that camphorol can attack in the human body. Those target genes that are associated with camphorol. That is what will be seen here with their gene ID. So all these genes are genes that are associated with camphorol. Now, if you are doing a study, you must be careful on the organism. Here, this is against Halianthus annus. So, but if you are working for human, you look for the genes that are expressed in human. So all these ones are expressed in plants. All these ones are expressed in plants. You look for the ones that are expressed in human. So maybe I can just type camphorol human. Maybe I want to look for genes. Camphorol human. If I type camphorol human, it may give me some genes of camphorol that are associated to humans. If they are none, it will not give me anything. But Pubchem is not useful.
Well, I didn't get what you said. Network was bad. You said if there's something that targets organism.
Yes, I was saying that outside looking for exact match, you can use Pubchem to get the genes. Here we help you know the genes that are targeted by the structure camphorol. Once you just click on genes, it will list. There are 19 genes here that are associated with camphorol. Those ones, these are the ones up that have been deposited in Pubchem. That doesn't mean that there are only 19 genes, but for the ones deposited in Pubchem, there are 19 genes in Pubchem that are associated with camphorol. Now, it means that camphorol can attack those genes, but you need to now ask yourself: these genes, are they the genes in human body? Some of them are genes in plants. Some too are genes in human body. How will I now know? If you look at taxonomy, like gene ID, this is gene ID for this gene, the gene symbol. The taxonomy is showing common sunflower. So this one is not for human. This one is for sunflower. So that means if you are working on, if you are looking for the genes that are expressed, that are targeted by camphorol, you search for the ones within human. These are different plants. You search for the ones within human. You may not really find anyone for human. You may still find, depending if it has been deposited in Pubchem. But Pubchem is not ideal for bioinformatics, because if you are looking for biological data, Pubchem is not ideal for bioinformatics. They just included those genes there just for us to see that those genes are associated with camphorol. Then here is protein. These proteins are the active site, disease active site in the body. So that there are 8 proteins that are associated with camphorol. So once you click on the protein, you will see the proteins. Now this protein comes with accession code. In bioinformatics, protein has a code. Not just protein, but when you have a gene that has been sequenced, a sequenced gene comes with accession code. But proteins themselves has a code, which is the protein data bank code, PDB code. But sequenced gene, so these proteins here are representing sequenced gene, and this is the accession code of that protein. And this is the taxonomy, which is the organisms expressed. So if you want to know the proteins associated with the compound, you can come to this place, but make sure you look for human. Make sure you look for human. That is for protein. Then the pathway here is metabolic pathways of camphorol. The possible pathways. So these are the informations you can get in a structure. The possible pathways. But all these ones is not ideal in Pubchem, because in chemoinformatics what we are really after is chemical informations. But when you are looking for biological informations, then we'll go into bioinformatics. But here, these pathways, there are 62 pathways, and these are the pathway: diglycoside biosynthesis. So it all depends on your interest. If you want to know if camphorol is taking the possible metabolic pathways of camphorol. But then, what is my major interest here? Okay, I will come back. Okay, basically, let me look at bioassay. This bioassay,
Practical.
this bioassay talks about experimental work, biological assays in vivo experiment or in vitro that has been performed on camphorol and their results. So maybe you want to look at some work that has been done on camphorol. Once you click on bioassay, you will see here drug level treated with camphorol after 4 hours with liquid chromatography analysis. So that is the work. So bioassay talks about in vivo or in vitro works that has been published, works that are related with camphorol that has been published either in vivo, in vitro, or in silico. Once you click on any one, you will get the full details. Here is a TP transporter increasing deromycin intracellular accumulation. So these are works that has been performed on camphorol. This kind of work will help you when you are working on camphorol. You can see some published work that has been done and the results of those work. Here is ratio of camphorol IC50 to compound IC50 for human, and so on. So that is about bioassay. You can just click on any one of them. Inhibition of his target active site. So when you click on any one, you will see the results of such work, the detailed information about the work, the title of the work. This is camphor, a potential cytostatic and cure for inflammatory disorders. So these people, that was the work they performed. You see the results. Their results will also be published there. So you can see that you can use Pubchem to get any kind of information you want to get about a, a compound. Maybe you you discovered a new compound and you are jubilating that you have discovered a potential drug candidate. You can come to Pubchem, do your search to see if a lot of work has been done. If no much information has been done, that means you have actually gotten a novel compound. So this is showing the possible result. Now this is the data table, and is showing the activeness, the IC50, which is the minimum inhibitory concentration at 50% of camphor against inflammation. And at this place you will see the target, the gene that was used, and the protein, ribosomal protein 56 kinase alpha 3, and it was for Homo sapiens on a human. So this is information about that work that was published. These are the informations you may need. So that is if you are working on bioassay. If you want to look at the biological work that has been carried out on your compound, you can browse through here, and this will even help you to know what that have been done and what that has not been done, so that it will help you decide topic if you are working on plants. So if you are working on if your research is based on compounds, if your research is based on a compound. The first one I explained was if your research is based on plants. If you have a question, you can do well to ask. Hope you can hear me. Hello. Can you hear me?
Hello?
Okay, it seems network is really bad. But then, where did you stop? Like, where did you stop? Let me know.
You are still doing something on the bioassay.
Okay, that means we are on track. So I was just showing you. Uh, I was just showing you what can be done. What can be done on the bioassay. You can use
Can you
it.
hear me?
I can hear you. So
Yes.
you can use it and search for literatures related to your compound. Because let me just give you a hint now. Sometimes if you work with a plant, let's say you work with scent leaf. Hope you can hear me. Can you hear me?
I can hear you.
Okay. So let's say you work with scent leaf and you discovered about 150 compounds in scent leaf. I want to see if scent leaf can be used to treat maybe if scent leaf can be used to treat malaria, and you did a work of those 150 compounds, and you got a compound, you got one compound A, that is more effective. You now have to concentrate on that compound. So now your work now is no more plant based but structure based. You now look at the structure of that compound and see how you can use that compound, how that compound now can further treat malaria. Maybe you can ask, look at that compound and see if some work has been done on that.
I can't see the screen. I can't see the screen.
Okay, let me share the screen again. All right. I'm sharing back the screen.
Okay.
In case you see it, let me know.
I'm seeing it now.
All right. So as I was saying, you will now concentrate on camphor and come to bioassay. Under the bioassay, you can now see works that have been done on.
What kind of rubbish is this?
I have a network challenge.
Honestly, I don't know. Yeah, it's just busy. They'll continue.
Okay.
It is frustrating me.
Yeah, sorry. So you will now see some works that have been done on camphor, some related work that has been done under your plant, under your compound that gave a very good result. So that is for bioassay. Then literature, there are about 9,565 literatures. They are related to camphor. So these literatures are works published that has something to do with camphor. So they are literature. Some of them are just peer review, and some of them are. But bioassay is mostly for experimental work. But this literature may contain some experimental work and non-experimental work, peer review journals related to Pubchem, related to your compound, the date of publications, the source, the journal source. You get them here from Pubchem, mostly the PubMed source. If those journals were published, we are linked or we are indexed in PubMed, you get the PubMed ID, the journal type, if it's article or just a review work, and the date of publications, then the name of the journal. So this is basically to deal literatures. So let's say you are working with a compound and you want to deal materials on that compound, just come to Pubchem. You can get the literatures of those compounds to help you build your work. Patents, these are people who have registered who have gained their license on Pubchem. So maybe people who on the camphor, maybe people who discovered novel results that has not been done with the camphor, and then they went for patents. So you see that there are about 538 patents on camphor. So it is not a novel compound. It is not a novel compound. So these are some informations you can get. It all depends on the researcher's interest. What do you want to do? What do you want to do? Then what do you want to search? You can browse through it to know what you want to do. But then all these ones as for exact match. All these things you are seeing now are under exact match.
Man.
Yes. Let me now come to similar structure. You see, similar structure is to get those structures that have the same scaffold. Those structures that has the same scaffold with camphor, like structures that looks like they have the same parent chain with camphor. That is what you get under similar structure. You want to get structure that looks like camphor. Maybe camphor has been active, and I want to now get other structures that have the same parent chain with camphorol, probably those structures might be better than camphorol. You can do that with your Pubchem. Just click on similar structure search. Now when we come to similar structure search, there are 4 different kinds of information you can get under similar structure search. The first information you will get here is the identity. Identity 1, meaning that there is just one compound that has that camphorol, which is this one compound. Then similarity. This similarity is different from substructure. Similarities are compounds that look like camphorol, and there are more than 1,000 compounds that look like camphorol. So do you want to work on compounds that look like camphorol? If that is your aim, you click on similarity. Now under similarity, all these compounds you see here now, they look like camphorol. They look like camphorol because if you look at them, they all look like camphorol. Yes. So but what happened here is just 13%. If you click search all, if you choose search all, it will search 100%. And so this 100% will show you all the compounds that have similar structure with camphorol. You can download all once. So you can now download all those structure once and look at the results. They are about 7,567 compounds that have similar structure with camphorol. So once you come to download and you want to download them, you come to download. On the download, if you choose SDF, all those compounds will download once as a single file, and it will take time. They will download once, they will not download separately because they are downloading once as a single file. Let me just show you. Let me click on SDF. So as I've clicked on SDF, you will see that it's telling me downloading. So it's downloading them. It will take time, but it will come as a single file. So all the 7,000 compounds that have similar structure with camphorol will be on a single file. So let me just cancel it. So now it means you are not going to do a work with 7,000 compounds. That one now they will download once. They are not downloading one after the other. You will not use your hand to download them singly. They will download once, and it is good because by the time you work on that 7,000 files, 7,000 compounds that are similar to camphorol, out of these 7,000 you may end up getting ones that are better than camphorol for a particular target of your choice. So if you want to work on similarity will show you compounds that are similar to camphorol, compounds that have similar functional groups, similar parent chain with camphorol. Then substructure here is they are greater than 1,000 compounds for substructure. Substructure means is exactly camphorol, exactly camphorol, but some substituent will be added, but they will maintain the same shape with camphorol. But similarity search will not bring out compounds that has the same shape, exact structure with camphorol. Similarity search will bring compounds that has similar structure. It may not be complete camphorol structure, maybe similar functional group, similar scaffolds, but substructure, standard substructure search. So it look for structures that maintains camphor. If you look at all these structures, they still maintain that exact camphor with some other substituents being added to it, but they still maintain camphor. That is a substructure, and we use this substructure more when we are doing, when we want to look for structural activity. Maybe as camphor is anti-diabetic, can, if you have that other structures that has camphor but with small functional groups added to it, let's see if they will still exhibit anti-diabetic property. So we use substructure, and here is just 5% search. 5% search got 1,000 compounds. If I want to do search all of them 100%, let's see how many compounds are there. There are 21,320 compounds that has the same structure with camphor, but the difference is some substituents were added, and that is this thing now is what we call lead optimization. Is a process in drug discovery. Once you have a lead compound, you optimize it. You optimize it by adding some functional groups to it that may improve the bioactivity, or reducing some substituents, so that you can see if it will improve the bioactivity. So you can use all these compounds here as optimized compounds for camphor. I've been talking too much. I don't know. Do you have a question to ask? Any question?
No, no question. Then I wanted to suggest: is it possible to have a pre? No, no question. I wanted to ask: is it possible to have a pre-recorded video because of this bad network, so I can also go through it on my own and write out the question? I think it will make the class faster.
Okay. Okay. That means what I will do on my own is to get videos recorded before time. Okay. But let me still see.
Yeah.
But were you able to play the last video I sent?
Yes, I was playing it. With a lot of business, I was trying to play it, but that one was interesting. For this one now, I need to play it over and over again.
Yes. No.
To get acquaintance.
Yes. All our classes are recorded. All. So even this class now is recorded. So I will send it to you, so that you.
No problem. That's what I'm saying. If it's possible to have a pre-recorded of the class. Let's say next one is Friday. If it's possible to record it before Friday, so what we'll be doing is question and answer, and you'll be putting lights in some areas.
Okay. Okay, I will try.
Yes.
I will try, but that will be double work for me, but I will try. Let me see.
Okay, sir. Because if not for this network issue, because I'm even climbing nearly into my kitchen because of the network.
That is Nigeria. So these are just structures that still maintain the camphor structure with additional substituents, and we got 21,320 substructures. Then superstructures. I don't advise somebody to work with a superstructure, because superstructure shows the fragment. Superstructure shows the fragment, like parts, small parts that make up camphor. Like if I click on superstructure, what you'll be seeing there is fragment of camphor. Like look at what now you are seeing acetone, showing that camphor has a carbonyl. All those small fragments that make up camphor. they are what will be seen under superstructure. So we don't need it. It's of no use to us, but it's still there. So I don't usually use this one because I don't see a need for it. I wonder why it was there in Pubchem. Maybe people who has interest in that in superstructures might go for it. So now here is this 3D similarity. It's the same thing with this similarity structure, which is compound that looks like camphor. But here we only bring out 3D, the ones that have 3D, because only this similarity search will bring both 3D and 2D. But the 3D similarity search will only sort out compounds that look like camphor in their 3D form. In their 3D form. In their 3D form. So that is what that part does. Oh, it has been so heavy. But then we are not done with Pubchem. We are not still done with Pubchem. Now,
Yeah.
if you use Pubchem, you are going to reference Pubchem. Yes, when you use Pubchem, you are going to reference Pubchem because the informations were not from your head. You got it from somewhere.
Yeah.
So you are going to reference them. Let me show you. How we have Pubchem kept. They already kept. Their reference pattern. If you use Pubchem, how to reference them? I will show you. Okay. Now let me come back. If you used Pubchem, and you want to reference Pubchem, I'm hearing echo. It's okay. Maybe try to turn off your mic.
Okay.
Okay. So if you use Pubchem and you want to reference Pubchem, come to about. Once you come to about, then click on publication or citation guideline. Here you will see how to cite Pubchem. Depending on the reference style you are using. So if you are using upper style, you just copy this. You copy this and put in your work if you used Pubchem, because you must reference them. And they always update the citation. Like here it's showing 2025, but when I used it last year it was 2024, so that it will be a current citation. So if you're using upper style, which is most common to us, you just copy the citation and paste in your work. This is publications. Let me see what is under publication. So it's very important, because you cannot just use Pubchem and after using Pubchem you did not cite it. So that is very wrong. So this publication is not useful to us. It's showing journals about Pubchem, updated journals about Pubchem. My interest is on the citations. So you can copy the citation and paste in your work. Then lastly on Pubchem, what we're going to learn again in Pubchem is how to you can use Pubchem to look for compounds that has been investigated against a disease. Yes. Let me come back. Let me say I want to look for breast cancer compounds. I want to know compounds that has been studied for breast cancer. What I will just come and do, I will type breast cancer. Once I type breast cancer, it will give me compounds that have been studied for breast cancer. Compounds that has been investigated for breast cancer. So that is what is here. If I click on proteins, so let's see, I want to see compounds that has been studied for breast cancer. So if I click on proteins, so under proteins you will see studies on breast cancer and the compounds that we are investigated. So if I want to know compounds that has been investigated for breast cancer, if I click on this first one, let me select this one. So if I open it, this study on breast cancer, you will see some of the compounds that people have studied. This is breast cancer anti-estrogen resistance protein. So compounds that have been studied against this protein, you will see it under section 3, related. Okay, I'm coming. Okay, here does not show. There is no compound here. But I'm supposed to have related compounds, related chemicals. Okay, these are the proteins. Okay. This was at the interaction. Okay, there is no compound here identified in this place, so I will just go back. Let me try typing cancer deaths. Let me search for that. This is what I've done. Program deaths, like program the cancer deaths. I'll click on proteins. Can you hear me? So if I click on proteins, now this is programmed deaths. Now to show you something, let me click on this. So this is a work done on cancer, programmed cell death. You will see how many compounds that has been studied against this target. You see how many compounds that has been studied against that particular target. So this is chemicals and bioactivity. So all these are structures, compounds that has been studied against that target, and we have 2,250 compounds that have been studied against the program cell death. So another usefulness of Pubchem is to know compounds that have been studied against a target. So those compounds can be used as your control. Okay. Hello, are you with me? Are you still there? Oh. So So we can be able to see those compounds that has been studied against their 2,250 compounds that has been studied against program death, program cell death, program cell death. So that is what another thing you can get from here. So when you have a disease target, you can actually get some compounds that has been studied on that disease target. You look at the activity of that compound, like this place is showing you the activities of those compounds. You can compare them with your own. So these are most of the things you can do with Pubchem. When next we meet, we shall now basically look at ChemBL and see what we can get from ChemBL, right? So thank you so much. God bless.